Okay, so now let's talk about the repercussions of protein structure. Um, Particularly in changing protein structure and how it changes the interactions that you see. So, this first one we're going to look at is called mad cow disease. This is probably something that you've heard of before. It's also called the Creutzfeldt-Jakob disease. Really, what happens is the here you'll see that we have a normal prion and a diseased prion. So this is this particular protein. You'll see that there's a lot of alpha helices in the normal one. However, in the diseased one, there's a lot of beta sheets. So what happens is the prions have multiple tertiary folds. They have multiple specific folds that allow it to do that protein to do its particular job. However, when a diseased prion gets next to a normal one, it causes the normal one to change its overall interactions, and it changes it from that alpha helice to a beta sheet. And when that protein becomes beta sheet, it is symptomatic. So this is what causes mad cow disease. So things such as a spongy brain, or it destroys the cow's brain, actually. And then these changes in structures are transmissible. So one bad protein will go near another bad protein, have quaternary interactions with that protein, and it will change the structure of the normal prion into a bad one. So this is how mad cow disease works. It's just because of the change in structure. So the primary structure of this protein is the exact same. Nothing changed, nothing broken, came back together in a different way. The order of those amino acids are exactly the same. However, now they have different shape in correspondence to one another, and it causes a disease in cows. Another example of this is sickle cell anemia. So I learned a lot about this in like my high school genetics classes and stuff, but never learned why this happened. So you can see here in the picture, blood cells typically are this round O shapes. These look like this. So these are hemoglobin. So we talked about it earlier. So we have those 4 proteins all together. However, so if we look at the actual protein chains, and HB stands for one of the specific proteins in hemoglobin. So if we look at a particular portion of hemoglobin, so this is just a small portion that I'm showing you, the primary structure, there's actually a change in the primary structure here. We have an acidic amino acid in the normal protein. However, in a modified version of the protein, or technically the diseased protein, you have a nonpolar. The acidic and the nonpolar have very different tertiary structures. And so the beta chain without oxygen, so when your hemoglobin doesn't have oxygen in it, because it has a nonpolar amino acid there instead of an acidic amino acid, when there's no oxygen there to help it hold its shape, it changes its tertiary structure. So the overall structure then has to change, and this over time will cause polymerization of the hemoglobin, and red blood cells will begin to sickle. So you'll start to get a change in tertiary structure and you can't get it back. And that's why you see the sickled cells. And then the last thing is just talking about denaturing proteins. So proteins, tertiary structure. So what do I mean by denaturing proteins? This is a disruption of the forces that stabilize either the secondary, tertiary, or quaternary structures. So the primary stays intact. So we're not actually changing the order that the amino acids show up. We're just changing how they interact with one another. So for example, just in a visual context, an active protein, such as in this egg, it's going to have a particular structure with all of these alpha helix and beta sheets and everything, very particular structure to it. And then when you heat it, add acid, base, heavy salts, all these things we're about to talk about, you denature the protein. So you break apart that tertiary or secondary structure or quaternary structure. So how you can do this is heat. So when we cook food, the reason it changes colors or it looks different is because we're changing how the proteins fold. So with heat, you disrupt the hydrogen bonds and the hydrophobic or hydrophilic interactions. So this is how you sterilize things. So if there's proteins in a bacteria molecule or something like that, you denature that, killing the bacteria. And then in cooking process through these eggs, like I said before, or meat or something like that, you're changing that protein structure. Another way to do this is agitation. So I guess if you think about eggs, again, if you whip egg whites long enough, you're going to get this nice fluffy white stuff, right? That's very different. It's because you're agitating those proteins so much that they're changing their tertiary structure and secondary structure. You can also change the pH. So if you add something acidic, so like if you put vinegar with eggs, you actually cook the eggs a lot faster. So the charged residues become neutral. You break those salt bridges, again, breaking tertiary structure. And additionally, heavy metals. So this is why lead poisoning and mercury poisoning is dangerous. Adding heavy metals to proteins is going to interact with the R groups of those proteins, change the overall tertiary structure, disrupt disulfide bonds, denature the proteins. I don't have this on the slide. However, this is actually also with viruses. So we're disinfecting literally everything right now, right? Like everybody cleans constantly. Every time we go anywhere, we have to put alcohol on our hands in order to do our hand sanitizer and everything. That is changing the environment of the proteins that the virus lives in, and therefore kills all the viruses and bacteria that are there. And that's why cleaning products work. You change the pH of those proteins, making them useless. They can't do any work anymore. Kills everything pretty much. So that is a connection into our lives today as well.